Okay, so the next part of this chapter is about the special senses. We already saw before the general senses, the receptors, what type of stimuli do they receive, and where are these receptors located, how do we name them based on the stimuli that they receive. Now for olfaction, olfaction is the sense of smell, and it is brought into this region in our brain for the olfactory cortex, which is just below the frontal lobe, or well, it's in the frontal lobe in the lower area. So we have the olfactory nerve. So the olfactory nerve basically connects these neurons that we have in the olfactory mucosa, which is at the roof of the nasal cavity, where we have the olfactory mucosa. And over there we have receptors, chemoreceptors that receive odor molecules and bind them. Now these receptors have like a hair-like extensions, and then these extensions connect to this olfactory bulb that is located, or well, these cells extend an axon that connects into the olfactory bulb after these axons goes through the cribriform plate of the ethmoid bone. So we have around 10 million bipolar neurons in our olfactory mucosa, and these ones are actually replaced by some cells that we have in this epithelium every 2 months. And one of the things that researchers are trying to find is why this happens with these neurons, with these receptors. Because that can give them, if they find the reason why, that can give them a clue of then how can we help the central nervous system neurons to regenerate when they are damaged. So what is special about these neurons and that they regenerate, and then what is happening with the other ones that they don't? So research in this area is very important, because if they find out something from these olfactory neurons and they can transfer it to the other type of neurons, like the central nervous system neurons, then there are chances for the people who has injuries in their brain to recover much easier. So then once this olfactory epithelium binds to these odor molecules, they generate a receptor potential, and then this receptor potential travels through this axon of the bipolar neuron that goes through this cribriform plate, and then you have the terminals that releases a neurotransmitter that is going to depolarize the neurons that are located in the olfactory bulb. And then you saw that in the brain that you dissected the other day, where is the olfactory bulb. And then this olfactory bulb extends axons that goes posteriorly that will form the olfactory tract. And then remember that cranial nerve number 1 is known as olfactory nerve. And this actually bypasses these axons from this nerve, bypasses the thalamus. It goes directly into the olfactory cortex, so where we process the information that we receive, and then you connect that information to the limbic system for emotional responses. And then sometimes, of course, normal behaviors that are expected. So for instance, what if we started to smell something that is burning very close to us? Well, we'll exit the room, and then we start to hear the alarm, like the fire alarm, and then we know what to do. So sometimes this type of memories basically regulates your behavior too. So here is the view of this olfactory epithelium. It will be located in your nose. So this is a mid-sagittal section of the nose with the lateral view. So this is the hard palate. And then here we have the floor of the nasal cavity. Here's the roof. And then here are the superior, middle, inferior nasal conchae. And here's the vestibule of our nose. So when we inhale these odorants, if they bind into this part, you don't elicit the sense of smell. They have to go here into this olfactory epithelium, because that's where we have the neurons, the bipolar neurons, which are these, that has receptor for these odor molecules. And then again, they extend the axons through this bone, which is the cribriform plate of the ethmoid bone, to connect into this olfactory bulb, and then you extend axons that form this olfactory tract. So then if you magnify this part, so this is what you will see. So you will see these olfactory neurons, they are bipolar, so they have two extensions. So this and this. So this is the axon, and here we have the dendrites. And then we have these finger-like extensions, which are the olfactory cilia, which will be the ones that binds these odor molecules that we smell. And then you generate a receptor potential, and then that receptor potential forms an action potential that ultimately migrates along this axon. And then in these axon terminals, you release neurotransmitters that are going to activate these axons that are located in the olfactory bulb. So this is a magnification of the olfactory bulb. So here you have a synapse between these olfactory neurons and then these olfactory tract neurons. Along with these neurons, we have in this olfactory epithelium these simple columnar cells, and then we have some cells that produces mucus and some glands that produces mucus that actually is not shown in here but coats this epithelium, protecting these cilia, and also helps to keep the odor molecules for a little bit of time in there, so that you can have more chances of making some action potentials. Now, we have interspersed, or in between these cells, we have these basal cells, which are the responsible for replacing this epithelium when it's time to be replaced, like every 2 months. So any questions? Okay, so this is what happens for the function of these neurons. So if you magnify this tiny piece from the cilia, you will see the plasma membrane of the cilia. You will see some receptors, some enzymes like adenylate cyclase, and some channels, ion channels. Again, this is the phospholipid bilayer of one of these cilia. This is the cytoplasm. So here we have the cytosol, and this will be the extracellular fluid. So you will have then some receptors that binds to these odor molecules, and these receptors are coupled or linked to these proteins that are called G proteins. So once you bind this odorant to these receptors, G protein becomes activated, and then G protein comes and binds to this enzyme. Part of this G protein comes and binds to this enzyme, which is called adenylate cyclase. And one of the things that these adenylate cyclases do is convert adenosine triphosphate into cyclic AMP. So it strips away two phosphate groups. And then this cyclic AMP, which stands for adenosine monophosphate. So it's going to have only one phosphate group because you lose two phosphates when you use this enzyme. So then this adenosine monophosphate is going to bind to this inner part of this ion channel, so that this ion channel opens, and then you have calcium and sodium entering into the cells, into the cytosol. And since you have these positively charged ions that comes into the cell, what is going to be the result? Are the cells going to depolarize or hyperpolarize? So remember, in action potential, we have depolarization and hyperpolarization. So the cells typically are negatively charged. And then when you trigger an action potential, these cells have a phase of the action potential. So then, in this case, since you open these sodium channels, and these are positive, and then and calcium channels, calcium-sodium channels, and then these two enter into the cells, what is going to happen to the cell? Is it going to become positively charged or negatively charged? I'm going to sing a song. Say it loud, say it clear.
They would be negatively charged, right?
The cells in resting membrane potential. But then when you have these ions, calcium and sodium, look at the sign there, the plus sign, then enters into the cells will become so this is RMP, resting membrane potential. So that's the normal state of the cells. And then when you open channels, the cells will become what? Positively or negatively charged? Positively charged. So this is because you open sodium and calcium channels in that case. Open. So this is called what? Depolarization or repolarization? Depolarization. And then you reach the peak, and then the voltage goes down when you close sodium and calcium channels. And then you return back to negative, which will be what? Repolarization. So this is becoming positive and returning to negative state. So this is the action potential. Kind of crazy thinking that even for smelling, look, you need calcium. So calcium is not only for having strong bones and strong muscles, muscle contraction, but also for smelling. Calcium is everywhere, it's needed everywhere. So any questions? So that's the action potential. Then, and the title was there, olfactory cell depolarized with calcium and sodium entry. I didn't need to read, right? Okay, so that was then how the sense of smell happens. Now for gustation, which is tasting. So we have these chemoreceptors on the dorsum of our tongue and also in the pharynx, in the area above the epiglottis. So we have some receptors for tasting, and in the case of the ones that we have in the tongue, they are called taste papilla. So they are small protrusions, and these papilla, they have taste buds, which are actually the actual receptors for tasting. And we have three main types of papilla that has taste buds, and then we have one that it doesn't have taste buds. So the three types that has taste buds will be the fungiform papilla, which are like the most common type that we have in the tongue, and then it has very few taste buds, but they are distributed everywhere, but they are more likely to be found in the tip of the tongue. That's a fungiform. And they look like little red dots. If you look at your tongue after you brush your teeth and you brush your tongue, if you look at it under a mirror, it looks like little red spots. And then the circumvallate, which are also known as valid papilla, there are not so many. We have like 8 to 12 arranged in an inverted V shape on the posterior part of our tongue between the posterior third and the two anterior thirds. And these ones, they have many taste buds. Now, the foliate papilla, which we have basically on the sides of the tongue, they have taste buds, but as we age, we lose this ability to taste with those taste buds on the papillae that we have on the sides of the tongue. So it is said, I'm not sure, but it can be possible that some children that didn't like onions when they were little children, it is because they have the, in general, all the taste buds are, of course, as someone is young, have more ability to taste in children and adults. But these lateral taste buds, apparently they're more sensitive for things like onions. So hence, many of them, they don't like onions when they are children. And then as we lose these taste buds, and then you're an adult, you can still dislike onions, of course, but you are less sensitive to that strong flavor of onions. And then one advice that is given is just like, don't give these big chunks of onions to little children, because it's not only the taste for them, but also the texture that matters. So you can grind it and put a little bit in the food, and little by little they will assimilate. They will acquire that taste. Now, the other type of papilla that is actually the most abundant, but it doesn't have taste buds, will be the filiform. And that one has a particular function. It will be to detect the texture of food. I think I told you this before, but just in case that I haven't, they can take a piece of mango or strawberries or an apple. And then, of course, if you have tasted that type of food before, you will not only know how it tastes, but also you will know how it feels in your mouth. So they can destroy these chemicals that give taste from this little piece of fruit, and they give it to you, and you will still be able to tell what it is, even without the flavor, just by texture, because we have this filiform papilla. And then cats, my gosh, they have very dry filiform papilla. Mm-hmm. If a cat licks you in the skin, it feels like sandpaper. It's their filiform papilla that helps them to groom their fur. So here is the longitudinal section of a valid papilla, which is the largest type of papilla that we have. So we have this dome-shaped papilla, so it's like a magnification of this type of papilla. So this will be the epithelium, this will be connective tissue, and then you have the sensory neurons that connects to these taste buds. And then this will be a taste pore through which the molecules of food comes in, and then you can have binding of these chemicals in food or liquids into these receptors. So, location specifically, fungiform in the two anterior thirds of our tongue, most likely found in the tips and sides. That's the one that we saw before. It has a mushroom shape, and then it has moderate taste buds innervated by cranial nerve number 7, which is what cranial nerve? Facial. That's the facial nerve. And then circumvallate. This one, it is located in the posterior third of the tongue, arranged in V shape. They're large. Actually, these are the largest. And then they have many taste buds, help us detect bitter taste. Is innervated by, they are innervated by cranial nerve number 9, which is glossopharyngeal. And then foliate. These are located on the sides of the tongue near to the back. They detect sour tastes, and they are leaf-shaped. They are innervated by cranial nerves number 9 and 10. So taste from the two anterior thirds of our tongue then is detected by cranial nerve number 7, and then from the posterior part of our tongue, and also from the pharynx is by cranial nerve number 9, and then 10. So something about food texture is also important because it helps to enhance the taste experience or possibly ruin it, depending. Temperature of the food also influences taste perception and flavor intensity, because when you have, not to the point of scalding your tongue, of course, but when you have higher temperature, you increase the chances of binding these molecules into these taste buds. And then, so here we have the superior view of our tongue. So this is the dorsum of the tongue. So this is the area where we have a fungiform papillae. This is the area where we have the foliate papillae. And this is the area where we have the valid. And then scattered all here will be the fungiform. And then we have some taste receptors here in the epiglottis area, especially for water. Okay? Water has a taste. It tastes like water, and there are different qualities of water. But now for pain and temperature sensation, it is provided by B3, mandibular branch of the trigeminal nerve. And then if you like spicy foods, you are a masochist because that elicits pain. It's not a taste, it's just pain. So then the function of these taste buds, how it happens. Well, for salty taste, the sodium chloride, especially the sodium part, of course, will enter into the taste receptors. That will depolarize your cells. And same reason for when we saw the depolarization of the olfactory neurons. Okay, so you have opening of these channels, or the channels already, are open, and then you have depolarization. So you have a positively charged cell. And when you depolarizes these cells, that will open calcium channels. So again, then calcium is also needed for tasting. And then with that calcium that you have there, you increase the chances of releasing neurotransmitters, because that basically sustains a little bit more the depolarization, and then you generate your action potentials after this in your postsynaptic cells. And then for sour taste, you have hydrogen ions in foods, like acidic foods, that depolarizes these receptor cells. I mean, hydrogen ions are having a positive charge, and then another thing that can happen, you can block potassium channels. And that will lead to this depolarization, and then you have the release of neurotransmitters. For sweet taste, you have sweeteners and artificial or natural sweeteners like glucose that binds to these G protein coupled receptors that are found in the plasma membrane of these cells, and then that leads to closure of potassium channels. When you close the potassium channels, potassium doesn't exit the cells, and that causes depolarization. So depolarization can be caused by preventing, in this case, and in the previous one, preventing potassium from exiting. And that leads to the neurotransmitter release and also the synaptic transmission. So here is a close view of one of these cells and the group of cells in the taste buds. So here we have the taste pores. So the chemicals from food enters through here, and then they combine into these microvilli. And then you have supporting cells, just like as the olfactory epithelium and basal cells that replaces these taste buds. So then what happens at this level, at the cellular level, for the function of these receptors? So you can open sodium or potassium, or hydrogen ion channels, and then changes in ion movements, like if you have sodium or hydrogen ions, that will start depolarization of the cells, which leads to opening of these calcium channels, and then you have the release of these neurotransmitters that are found in these synaptic vesicles. And that will lead to a postsynaptic cell to bind these neurotransmitters, and that will lead to the generation of an action potential. So that's for salty and bitter. Now for the sweets, again, it's just blocking these potassium channels, and then you can have depolarization of the cells. And then these postsynaptic receptors, they will send the taste signals from the papillae into a nerve that is called the chorda tympani of cranial nerve number 7, that is located inside our tongue. And then that exits the tongue, follows the pathway of cranial nerve number 9 and 10, and goes into the medulla oblongata, into a nucleus that is called the solitary nucleus. And then from there, you have some axons from this area that connects into the gustatory cortex in the insula. Questions? So here is then the pathway of this action potential. So you can have some action potential generated in the pharynx, in the posterior part of the tongue, the anterior part of the tongue, and then it's migrating to different nerves, 10 posteriorly, intermediate, or in the middle, number 9, and then anterior for cranial nerve number 7. And all of them goes into the medulla oblongata, into the solitary nucleus. You connect with these axons from the solitary nucleus that brings this information to the thalamus and then into the primary gustatory cortex. Now for vision. So we have accessory structures that protects our eyes, including the eyelids, which are also known as palpebrae. We have two. They connect laterally and medially into these commissures, and we have this action of the eyelids, which is basically closing and opening, so that we can sweep away the debris that might enter into our eyes together with the tears that we produce in the lacrimal glands. And then we have the conjunctiva, that it is a very thin membrane that is located anteriorly in our eyes, but also covering the internal part of the eyelids. So we have something that's called palpebral conjunctiva, and then we have that it's located in the eyelids, in the internal part of the eyelids. And then we have the bulbar conjunctiva that covers the part of our eyes, which is the area that is not colored. So basically covers the white part of our eyes. So this conjunctiva protects against microbes and provides lubrication. Now the lacrimal apparatus, which is made by the lacrimal gland, which is located superiorly and laterally in our eyes, these lacrimal glands produces the tears, and the tears by gravity, if someone is standing, of course, it will go down and then it tends to go to the middle of our eyes. So then we have lacrimal puncta located in the inner part of our eyes that is helping to drain the tears, because he has like little openings that helps to bring these tears into the lacrimal canaliculi, that is going to drain into a dilation of the nasolacrimal duct, and then from there our tears will go into the inferior meatus of our nasal cavity. So we have, besides that, we have the eyelashes, we have the eyebrows, and then this is a superior eyelid, the inferior eyelid. They connect both of them in the medial commissure and the lateral commissure. And then we have a raised area here, which is called the lacrimal caruncle. And then inside there, that's where we have the lacrimal puncta that receives the tears as they flow from superior lateral into inferior medial. So that's the flow. And then this will be the bulbar conjunctiva And then this part that covers our color part of our eye will be the cornea. Now, within these eyelids in the edge, we have not only these eyelashes, but also we have openings for some glands, sebaceous glands, that maintains these edges of the eyelids lubricated. This is a view of the lacrimal apparatus. So here we have the lacrimal gland that drains the tears, and then the tears goes into this caruncle where you have the lacrimal puncta, and then this lacrimal puncta drains into this canaliculi and then into the nasolacrimal duct, which has a dilation here, which is called the lacrimal sac, and then you have the drainage under the inferior nasal conchae. So this is an X-ray view of the lacrimal duct with dye injected to see if you have any obstructions. In this case, this is a narrowing portion. It's supposed to be more dilated. For extrinsic eye muscles, you already saw them before, but we're going to see them again. We have 6 muscles that connects into the outer layer of the eyes, which is called the sclera. So we have 4 rectuses. Superior, that help us to look up, inferior, that help us to look down, lateral, that help us to look to the sides, medial, that help us to look medially. So those are the 4 rectuses. And then we have the superior oblique that runs like this, and then we have the inferior oblique that runs like this. Superior oblique help us to look down and out, inferior oblique up and out. And they are innervated majorly by cranial nerve number 3, except for the lateral rectus, which is innervated by cranial nerve number 6, which is abducens, and the superior oblique, which is innervated by the trochlear nerve or cranial nerve number 4. So here you see the functions of each of these extrinsic muscles. So this is the view of all of these extrinsic muscles. So this is superior rectus, this is inferior rectus, lateral rectus, and medial rectus is in the other side. And superior oblique is supposed to be here, inferior oblique in here. And then this muscle above this superior rectus is the levator palperis superioris, which it doesn't connect to the sclera. So this is the sclera of the eye, but it connects to this tarsal plate, which is basically cartilage and connective tissue from our eyelids, the superior eyelid. So this one is as well innervated by cranial nerve number 3. So this one helps us to open and close our eyelids. And then here, this one is the bulbar or ocular conjunctiva, as you can read there. And here, this will be the palpebral conjunctiva. And then we have the orbicularis oculi muscle that help us to close our eyes. So part of this function of the muscles, and then we have intrinsic muscles, help us to protect the retina against damage. So for instance, with these intrinsic muscles, I already told you about this before. So we have in our iris a couple of muscles. So we have the radial muscles and the circular muscles of the iris. So what they do is either open or close the pupil, so that we can regulate the amount of light that goes into our retina. So if you were looking anteriorly to the eyes, and this was the iris, we have a muscle that goes medially, so this is the pupil. So this muscle that it's located like this in a circular fashion, this one, it is the circular muscle. And then we have another muscle that goes like that. So this is the radial muscle. These muscles are having different function. So the radial muscle is innervated by sympathetic nerves. And this circular muscle by parasympathetic nerves. In this case will be cranial nerve number 3. So, actually, am I doing it the other way? No, it's fine. Okay, so what would be the result of someone being scared, facing a dangerous situation, and activating the sympathetic nerve? What will happen with the pupil? Will become dilated or constricted? Dilated. When someone gets scared, they open their eyes widely. Why? Because you contract this muscle, so that makes your pupil to dilate. That is called mydriasis. And then, if someone is activating parasympathetic system, cranial nerve number 3, these muscles relax, and the circular muscle contracts, the pupil becomes smaller, so that is called miosis. So that's happening with cranial nerve number 3. You have miosis. And this happens because someone enters into a room, it's very bright, so you don't need as much light to see. In that case, you constrict. Much better than my drawing. So look at the arrows. Increase light intensity, decrease light intensity. Normal light intensity, you have particular diameter in your pupil. So this is the pupillary sphincter muscle, or the circular layer. So it is in balance with the contraction of this radial muscle, which is called dilator muscle. Then when someone goes into an area where you have increased intensity of light, this parasympathetic system makes this to constrict. See how the pupil becomes smaller, and you can see better the this muscle in here, which is the dilator muscle. And then the opposite, when you enter into into a decreased light room, so your pupils become larger because this sympathetic system kicks in, so you will have relaxation of this muscle and contraction of this muscle. cat's eyes during nighttime. Very big, the pupil. Okay, now specifically for our eyeball, so we have 3 layers. We have the fibrous tunic, which is called the sclera. And then the sclera is called sclera because it's very thick, it's protective. And anteriorly, this sclera, it becomes an epithelium that it is made out of several layers of cells that is called the cornea. And the cornea is very important to focus the light into the particular area of the retina that has more photoreceptors. So that's the outer layer. So it's made by the sclera and the cornea. And then the middle layer, which is called the vascular tunic, includes the actual pigmented epithelium and the blood vessels. So this is called choroid. And then we have, posterior to our iris, a structure is called ciliary body, where we have some muscles and also we have some ligaments that are connected to a biconvex structure that looks like this, that is called the lens. And that lens can become flattened or thicker. So this ciliary body helps in lens accommodation so that we can see when the objects are coming nearby. And then we have part of the vascular tunic, the iris, which is the actual one that helps us control the size of our pupil. And then the most internal layer of our eyes will be the neural tunic or retina, in which we have photoreceptors, cones and rods. Cones help us to see in color. So we have colors to detect red light, then we have for blue, and then we have for green. And then the cones help us to, the rods help us to see black and white colors. So here are the layers of the eye in a mid-sagittal section. So this is the most external layer. This is the sclera. That's where the muscles, the extrinsic muscles connect. And then anterior, this sclera becomes the cornea. The cornea, it's an epithelium that focuses the light into the area of the eye where we see the most, that is called the macula lutea. 2/3 of the light, it is refracted or bent so that we can see better with this cornea. And then behind the cornea, we have part of this middle layer, which is the vascular tunica. So we have the iris, which is this one. And then we have the ciliary body with these suspensory ligaments that connects into the lens. So the lens brings 1/3 of the light into this area. So this one has more power of refraction than this. And then here, of course, we have the pupil. And then this part is called the anterior chamber, while this one is called the posterior chamber. And then here we have the posterior compartment of the eye with the actual choroid, where we have the blood vessels and pigmented retina that helps to bring these photons of light into the neuroretina, which is the most internal layer. And then we have blood vessels that comes in and out that are called the central artery and vein of the retina that enters through this optic nerve or cranial nerve number 2. Cranial nerve number 2 is made by the axons that comes from the retina and then exits the eye in an area that is called the optic cup. That's where we don't see, that's where we don't have photoreceptors, so that's our blind spot, the optic cup. And then the area for retina where we see the most, it is called the macula lutea, and specifically in the middle, we have the fovea centralis. That's where we have most photoreceptors. So that's the area of sharpest vision. And then all the branches of these blood vessels distribute throughout the retina to provide blood supply to the cells. So we have then these chambers or cavities, like the anterior cavity, where we have fluid that is called the aqueous humor. And this is produced at a particular amount and absorbed also at a particular amount. And if there is problems with production and reabsorption of this fluid, a person can have increased intraocular pressure that can lead to compression of the optic nerve and blindness. It can be unilateral, it can be bilateral. Sometimes with surgery it can be corrected, but sometimes it cannot be corrected, and the person ends up blind. Runs in families, sometimes it's genetics. And then this fluid maintains the intraocular pressure and nourishes the cornea and the lens. In the posterior cavity or compartment, we have another fluid, but this is viscous. It's like jelly. So this is called the vitreous humor. This one helps to maintain the shape of our eyes, which is kind of spherical, and then it supports the retina. It keeps the retina attached into the middle layer, into the choroid. And then the lens is a biconvex structure that has a cortex, and it has some fibers that helps to focus the light into the retina through accommodation. I mean, you saw this before. So this is the anterior chamber, posterior chamber, and this is filled with this aqueous humor that it is produced here, recirculates, and it is reabsorbed here in this area between the cornea and the iris. So we have some venous sinuses that absorb this fluid. So here we have then in this anterior cavity this aqueous humor circulating between the anterior chamber and posterior chamber, and then in here in this posterior cavity we have the vitreous humor that helps to maintain the shape of the eye and presses the retina against this choroid, keeping it in place. Then the blind spot, again, is or optic cup. It is where we don't have photoreceptors, so no image detection happens in here. And then the macula lutea is a small yellowish region. Macula means spot, lutea means yellow. That's where we have higher density of cones. So that's where we have the central vision and best color perception. So this is a fundoscopic view of the retina with an actual ophthalmoscope. So here we have the optic disc. This is the central artery and its branches emerging to provide blood supply to the different regions of the retina. And in here we have the fovea centralis in the middle of the macula lutea, which is on one side of this optic disc. So area of no vision, no photoreceptors, area of sharpest vision, because we have the greatest number of cones. And sometimes some people, as they age, they start losing some of these photoreceptors. It's called this problem macular degeneration. And then, of course, we have visual problems. So we will do this test in the lab, but we have then this test for blind spot, in which you will be bringing a chart to see when you lose vision. The retinal cells and their function. So we have, again, the photoreceptors that detect different colors. Like we have S cones, which detects blue light, M cones, green light, L cones, red light. And then we have not only the cones, but we have the rods that takes tones of black and white. And then we have these ganglion cells that actually will be the ones that form the optic nerve that receives input from bipolar cells. Bipolar cells receive the signals from the photoreceptors and then act as a bridge to connect to these ganglion cells. And then we have in between, in an area between these cells that is called the multiplex part of the epithelium. So we have some cells that are horizontal cells and another ones that are called the amacrine cells. So these horizontal cells help to connect multiple photoreceptors so that we don't see only one type of color tone. So we see the multiple color tones, because as the photons of light hits our photoreceptors, we can combine the different information and make sense of what tone of gray or blue and all the combinations that we see. And then the amacrine cells modulate information between the bipolar and the ganglion cells. So here is a magnification of this layer of the eye, which is the retina. So here we have the choroid. So this is the middle layer. And then here we have the pigmented layer of the retina, which is this. So this is the choroid, this is the pigmented area. And then when the light comes and shines here, these pigments keep the signal here for a while these cones and these rods that are here, so these are the rods, these are the cones, have greater chances of receiving this signal, and then they can transmit that information into these other cells. So then again, we have in this layer, we have the photoreceptors themselves. So here will be the cones and then the rods, and then they have a body, and then they extend this part of the neurons into these other cells will be the horizontal cells, which are these. And then these horizontal cells will send the signal into these ganglion cells, which are here. So this is the nucleus of these ganglion cells, and then they extend the axons to form the optic nerve. And then you have here the horizontal cells that connect different photoreceptors so that we can make sense of what is going on. So now if you see better details of these cells, the rods and the cones, of course, the shape itself tells you why they call this cone, because this one looks like a cone. So this will be what is called the outer segment. This is the inner segment where we have mitochondria. This is the cell body, and this will be the axons with the synaptic terminals, which you can see in this micrograph. So these are the cones, these are the rods. And then inside this outer segment for rods, we have discs. And inside this disc, if you magnify one of these discs, you can see this plasma membrane. And then you have these proteins that are called rhodopsins. So you have a retinal part, and then you have an optic part. This comes from vitamin A, and then when you receive a photon of light, this is retinal becomes transretinal, exits this opsin, and then it recycles back into what we call photobleaching, and then that is what it keeps these signals being produced inside these cells, and then you start generating your action potentials. And now here we have the different wavelengths that each of these photoreceptors can catch. So here we have between, and this is in nanometers, so we have between 350 nanometers and 500, the blue cones. And then we have some overlap of these blue cones with the red cones. And then you can see the different wavelengths. But out of this, basically, it's what we make this combination of colors. So, for vision. So when you have light coming into your eyes, first thing that happens is that, of course, you hit the cornea. And then the cornea will refract this light into this lens, and then the lens will send it into these photoreceptors. So you have then the light going into your rods and your cones, and then they will convert this light signal into an electrical signal. So then the rods will help detect dim light and motion, while the cones will detect color and fine detail. So then these electrical signals from these photoreceptors will be transferred into the ganglion cells by the bipolar cells, which will be sending that information and integrating that information into the optic nerve. And then the optic nerve will make a synapse in the lateral geniculate nucleus in the thalamus. And then from there, you will send the signal into the brain's visual cortex, which is located in the occipital lobe, through these optic radiations. So here is the superior view of this visual pathway. So this is the visual field. So we have the right, we have the left, and then we have the focal point here in the middle. So the temporal field on both sides is going to hit the nasal retina. And then the nasal retina will send the signal into the opposite sides. So the nasal retina from the right side will cross into the left, and vice versa in the optic chiasma. Now the temporal, the middle field, which will include binocular vision, will go into the temporal retina on each side. And then the temporal retina will carry that information unilaterally, so it doesn't cross. It continues in the same side, which is called ipsilateral. And then you will have these synapses in the pretectal area on the midbrain, and also in the lateral geniculate nucleus in the thalamus, and in the superior colliculus of the corpora quadrigemina, so that you can have reflexes in case that this is necessary. So you can move away, for instance, if someone shines you with light on the right side, you can close your eyes and move away from that. Okay, so that's why we have these connections. Now, in the case of this lateral geniculate nucleus in the thalamus, it will process this information, and then it will create these optical or optic radiations that goes into the primary visual cortex, in the occipital lobe, and then that way you can interpret what you will be seeing. And now for the cellular level. So these four receptors, once they become stimulated or not, they will have a change for release of neurotransmitters. In the case, in the dark time, you will release, after this photoreceptor was depolarized, release glutamate onto these bipolar cells. And these bipolar cells will be inhibited because glutamate is an inhibitor neurotransmitter, and then you will have activation of this ganglion cell. So see the firing rate, it is not as much. Now during that daytime, these light photoreceptors will hyperpolarize, and it will prevent the inhibition of this bipolar cell. So this bipolar cell will release neurotransmitters into this retinal ganglion so that you increase your action potentials, and that will send these action potentials into your optic nerve. So we receive more information during daytime than during nighttime, which explains why we have less action potentials during that nighttime than during daytime. And then for focus, basically, something that happens is that when you have your eyes and you have an object that it is farther away, your lens will be flat. And then let's say that this tree, and you're looking at this apple on the tree, it is 20 feet away from you. So ideally, you will have this light passing through the cornea and then into the lens and being focused here into the fovea centralis in your macula lutea. But then let's say that this object, as you are moving away, it becomes at a greater distance than 20 feet. In this case, the rays of light will become dispersed. So that will basically put rays of light away from this area, and that makes it to be blurry as you move away. Now let's say that you get an apple and then you're moving it closer to your eyes. So in this case, you will have some changes in your eyes to bring the rays of light into here. So your lens becomes thicker, become more rounder, because you'll have to basically relax these muscles of the ciliary body, and that will help for this to be a little bit more rounded, and that will help to bring the rays of light in here to refract them. And something that also will happen, it is that your eyes become medial, so you will be looking more towards your nose as this object gets getting closer. So this is called accommodation. While without this to be round and without looking medially, so you look straight ahead, this will be called emmetropia. Now, some problems of vision can be related to the curvature of your cornea. If your curvature of the cornea is not as it's supposed to be, you will have something that is called astigmatism. So you have an irregular cornea. And then this cornea will be divided into meridians, like if it was the earth, and then some areas will be affected, some others won't, but that's when they use these devices to know exactly where the irregularity happens. And then typically we have, no one will have a perfect cornea, of course, but we have like something that is called diopters, which is 0.5 to 0.75 in most people. And then in someone who has 1.5 diopters or above will need correction. And then in the case of this problem, you use cylindrical lenses to compensate for the uneven curvature. And then if you opt for it, and then, if you have the means, you can have LASIK, which will correct the astigmatism problem. Now for the ears, so we have a subdivision of the ears into outer, anything that is external to the tympanic membrane, and then anything between the oval window and the tympanic membrane will be the middle ear. And then we have the inner ear that is located internally to this round window. Functions: the outer ear catches the sound waves that are in our environment to bring it into the tympanic membrane so that the tympanic membrane vibrates. And then in the middle ear, we have 3 auditory ossicles, which is malleus, incus, and stapes, from lateral to medial, MIS, malleus, incus, and stapes. So they start vibrating and they start amplifying these vibrations so that we can transmit this into a series of membranes that we have in the inner ear so that we can move these hair cells that we have in the inner ear so that we can depolarize the nerves and then we can hear. Now in the inner ear, we have the vestibulocochlear apparatus. The cochlea is for hearing, and the vestibule is for maintaining sense of balance. Part of the external ear will be the auricle or pinna. This is the lobule. This is the external auditory canal that ends here in the tympanic membrane. And then we have cartilage here that helps to maintain the most external part of the external auditory canal open. And then here we have the temporal bone. And then in the middle ear, which is this, we have malleus, that it is connected into the tympanic membrane, the incus, which is connected to the malleus, and then is connected to the stapes medially. And then the stapes has a plate that closes the oval window. And then this is the inner ear embedded into the pitcher's part of the temporal bone, where we have this snail-like apparatus, which is called the vestibulocochlear. So this is the vestibule part, and you have the vestibular nerve connecting into here, which is part of cranial nerve number 8. And then we have the cochlea, which it has the auditory cells so that we can listen. And then we have here the vestibulocochlear nerve. And then the semicircular canals, part of the vestibule, help us to detect motion, like circular motion, angular motion, and linear acceleration. So we will leave it in here. Any questions?